All right. Hello, everybody. Welcome to AP review number 5 or 6, one of them. And what we're going to be talking about today is transcription translation and gene regulation. So we'll do transcription translation first, and then we'll do prokaryotic gene regulation and eukaryotic gene regulation. This one's going to be kind of long, and gene regulation in specific is kind of difficult, so I'm going to go kind of slow on those. But it's a pretty important topic. So what you're seeing on the screen here is the basics of transcription translation, which, if you remember, transcription and translation is about going from our DNA and transcribing that DNA into RNA, specifically mRNA, and then translating that mRNA into a protein. So this whole process of transcription and translation is how your body takes the DNA that it has and produces proteins. Because remember, you are a giant sack of protein, and so in order to make those proteins, we have to read your instructions of your DNA to make those proteins. And this is sort of like a very basic walk down of we do transcription first and then we do translation. And what you're seeing here in this picture is the prokaryotic cell up here and the eukaryotic cell down here. The main difference being that the eukaryotic cell has a nucleus. Okay, prokaryotes don't, and because of that, the DNA just sort of is able to float around freely inside the cell. And the only difference is that as the DNA is getting transcribed, because there's no nucleus, we can translate almost immediately. So the lag time is pretty low for prokaryotic cells. Whereas for a eukaryotic cell, we have to fully transcribe, and then that has to go through processing before the RNA can leave the nucleus and it gets transcribed. But we'll talk about that a little bit later here in a second. So here's the basics. Okay, for transcription to occur, what we're going to do is we're going to start with a segment of DNA. Okay, so here is my DNA. Man, this is really artistic DNA. Now, remember that a gene is one segment of DNA. So this whole thing could be one gene. Okay? And in this gene, it could code for multiple different proteins. But just for simplicity, we're just going to say it codes for one protein. Now, part of that gene is, the gene is broken up into basic parts. Okay? The first part is basically the beginning part. It's what's called the promoter. Okay? And the promoter does exactly what it sounds like. It promotes, meaning that it is promoting the beginning. This is the start. Okay, so the promoter is the start. So what the promoter does is it tells the enzyme that does transcription, this start here. This is where you begin. Okay, now remember, we are making mRNA. Okay, I'm going to do this in blue. mRNA is being made. So the enzyme that makes mRNA is RNA polymerase. A is because it's an enzyme, polymer because it's making something, and RNA because the thing that this enzyme is making is RNA. Okay, so we've got our promoter region. That's where we know how to start. Okay, the next part after that is going to be our sequences, right? Our gene sequences. That's definitely not how you spell sequences. And these could be really short. These could be really long, right? It kind of depends. It's just the part, once we start, we just read the whole sequence, okay? And what's going to happen is this DNA is going to sort of open up, and it's going to go, sort of go out and sort of create like a bubble, right? Where, this is a really inefficient way to do this. You get the idea. So it makes this sort of bubble, right? It unzips the DNA and unwinds it and creates this bubble where we have our bases on both sides. And then it's going to go through and it's going to read those bases and it's going to make an mRNA strip. So the general rule is that, A matches up with T and C matches up with G. Okay, but because we're going to RNA, this is DNA, but because we're going to RNA, well, what we're going to have is our A is going to match up with U, and our C is still going to match up with G. I'm just now realizing that you can see none of that. There we go. So our A's can match up with T's, our C's match up with G's, but because we're going to RNA, our T's are replaced with U's. So we're going to go through and we're going to read that sequence. So our RNA polymerase is going to come through and it's going to read a sequence of DNA, and from that, it's going to make a strand of mRNA. Okay. And that is once it's done reading the entire gene, it's going to get to a termination site, and the termination site, we'll just say it's right here. Okay. And the termination site is basically how the RNA polymerase knows to stop transcribing. This is the end. Okay, so this is the end point. And so what you're seeing here in this much more better drawn picture is you've got your promoter region in purple right here. And this giant green blob is our RNA polymerase. And it's going to go through and you can see it unzip and bubble out the DNA. And then it's going to create this strand of mRNA from that. Now it begins here at the promoter region and it's going to read this whole thing until it gets to the termination site. Okay. And that's where mRNA is going to come from. Now, in order to do this, we have to unwind our DNA from chromosomes and get it to where it's just a strand of DNA. Okay, so keep that in mind for later. But this is basically how transcription works, right? And it's pretty simple. If we're in a prokaryote, we can just start immediately taking this mRNA and reading it to make our protein. But if we are in a eukaryotic cell, then what we have to do before we can make our protein is we have to send this mRNA outside of our cell from the nucleus to a ribosome. So here we can see, okay, we've transcribed our mRNA, we process it, and then we're going to send that mRNA outside of our nucleus to our ribosome, okay? And so once we've got our RNA and we send it to our ribosome, then what's going to happen? This is basically just the same thing. Promoter region, we read it, we get termination site, there's our RNA, right? That's all real cool. Okay, so once we leave the nucleus, then we're going to go to the ribosome. And the ribosome is where we do translation, okay? Now, the ribosome is going to read the mRNA in groups of 3. So let me give you an example here. So we said we were going to do this in blue. So we've got our mRNA strand, okay? And just for simplicity's sake, let's say all of this is all the nucleus. Okay, so now we're outside the nucleus, and we've got our mRNA, and I don't know. ACC GU GUA. Okay, cool. So we've got our mRNA. We're outside the nucleus. This mRNA is going to find a ribosome, and that ribosome is going to read the mRNA, and then it's going to match up the mRNA with tRNA called transfer RNA. And the transfer RNA are these blocks of proteins, and what their job is to bring an amino acid to the ribosome that matches up with our mRNA. So the mRNA, remember, we read the mRNA in groups of 3. Those are called codons. And the tRNA is going to be an anticodon. So it's going to be the opposite to to whatever our codon is. So it has to, the reason that is because it has to match up perfectly in order to get the correct amino acid that the code is looking for. It's sort of like if you're reading a word, you know what it means because that word has a specific meaning, right? If you switch out the word, it means something totally different. So this tRNA is going to match up based on whatever the codon is. So it's going to be the opposite. So if our mRNA is AUG, our anticodon is going to be UAC. And that codes for a specific amino acid. And then what this ribosome is going to do is it's going to take these amino acids and it's going to start chaining them together based on the different codes. So then it's going to go on to the next codon, read its 3, find a new transfer RNA, and then start to bind these amino acids together. Okay, and that's going to create a polypeptide, which is our protein. So it's going to go down and it's going to read this whole thing until it gets to a stop codon, okay, a termination site. And the stop codon is going to tell it to stop. So let's say that stop codon is right here. That could be let's just say it's UA, right? U is a stop codon. So let's say that it reads this thing. We've got 3 amino acids here, one for this codon, one for this codon, and one for this codon. And once we get to the stop codon, it says stop. We have now stopped and we have made our 3 amino acid long polypeptide. Okay. And that's translation. Now, again, what you're seeing here is like I said, a much more better drawn picture here. So this sort of blue and yellow blob, that's our ribosome. We have 3 sites where we can sort of assembly line our production of protein. And you can see our codons are being read 3 at a time. We are combining our amino acids up here with these different purple shapes, and this is our growing amino acid chain. Okay. Because the whole point of this is to make protein, and that's what we're making right here. Okay. Now, that's how we make proteins. That's where genes come into play. Okay. But now that we know how to do it, we kind of have to talk about how you regulate it. Okay. Because remember we said there's different ways that we regulate those different genes. Okay. But you have to understand the basic structure of a gene first. So if, this is your DNA. Okay. You have to understand that we have a promoter region. I'm just going to call it P. We have our sequence region. And then you have your termination region. Okay. And so let's start with prokaryotes first, because prokaryotes are the simplest. So in a prokaryotic cell, we have our gene here, and all of these enzymes are required to express these different genes to make something. In this case, we're making tryptophan. Okay, so the more tryptophan we make, the more it's going to come up here and it's going to shut these enzymes off so that our body doesn't continue to make tryptophan. So what that looks like is right here. So we can see that, okay, we've got our promoter region right here. Now what makes prokaryotes different is that prokaryotes have what's called an operator. And an operator is like an on-off switch, okay? So when the operator is unbound, when there's nothing bound to the operator, that means the gene is on. So this promoter region says, hey, RNA polymerase, start here. It starts to encode these genes and it starts to produce the protein tryptophan, okay? Now, once we produce tryptophan, this is going to keep producing tryptophan just until something tells it to stop, okay? And what's going to tell it to stop is the tryptophan itself. So we have what the operator does is it provides a binding site for a repressor. Now you can see here this repressor is not going to fit in this operator, okay? So what's going to happen is the more tryptophan that we make, here we go. The more tryptophan we make, it's going to, here we go. more tryptophan we make, it's going to bind to this repressor, activate the repressor, and then the repressor is going to bind to the operator. And once that repressor binds to the operator, it's going to shut it off, telling it, hey, don't make any more tryptophan. So that is going to deactivate our promoter, which means this gene is now no longer on, it is now off. Okay, so in prokaryotes, they can be turned, the tryptophan operon is an example of a gene that is always on that can be turned off, and the thing that turns it off is what it makes. The tryptophan that it makes is what turns off the this whole gene, so that we don't make too much tryptophan. We also have another example of a gene that is off until it is turned on. So this is the lactose operon, and the lactose operon is imagine this repressor is bound to this operon always. It starts there. But when you induce something into the body that needs this gene to be turned on, so what happens is when the body consumes lactose, when you drink milk or something, that lactose binds to the repressor and pulls away the gene. So the presence of lactose activates this gene and turns it on. And what that does is it now starts to make enzymes that break down the lactose. So once we make these enzymes are going to break down this lactose. And once that lactose is broken down, we now don't have anything to bind to our repressor. The repressor goes back, binds to the operon, and it shuts the whole thing off because we don't need it anymore. We've already broke down all the lactose. So this is an example of a gene that only turns on when you need it, right? We don't need this on all the time. We only need it when we're trying to break down lactose. Tryptophan is a gene that is on all the time to make tryptophan, but it turns off once we've made enough, okay? And that's pretty much how prokaryotes regulate their genes, okay? It's pretty simple. Now, eukaryotes regulate their genes a little bit differently, okay? Eukaryotes have a little bit more, have a couple ways that they'd regulate their genes, okay? So the first one is through this RNA processing that we talked about up here. So we said, okay, in transcription, you transcribe, you make your mRNA, but really it's called pre-mRNA. It's not finished mRNA, and the mRNA has to go through processing. Okay, well, what is processing? What does that look like? So the order of operations here is that once you make your mRNA, so we've got our mRNA here in blue. That's definitely not blue. So we've got our mRNA. Okay, so we have a blue strand, and then let's say we have a light blue strand, and then we have a okay. So what's going to happen here is that in order is the first thing we're going to do is we're going to put a cap. So the first thing we're going to do is we're going to put a cap. Second thing we're going to do is we're going to splice. And the third thing we're going to do is we're going to put a tail. Okay. Now, the point of the cap and the tail is basically the point of the cap and the tail is to allow this mRNA to leave the nucleus. This cap and tail is sort of like the transporting parts to allow this thing to leave. If the mRNA tries to leave the nucleus without the cap and the tail, it's going to dissolve and it's going to break away, right? mRNA is designed to only last a specific period of time. This cap and tail ensures that the mRNA can get to the ribosome without breaking down. Okay. Now that's the first part and the third part. The second part is we splice. Now, what's the point of splicing? So the point of splicing is that you have these things called exons and introns. Okay. The point of splicing is that splicing generally cuts out the introns of a cell and leaves the exons. Okay. Now, why do we do that? Why do we even transcribe these in the first place? Okay. And the reason is because it allows us to flexibly make different combinations of proteins from the same gene. Okay. So for example, let's say, just to put it simply, let's say these genes that we're coding for like letters. Okay. So let's say that we have, I don't know, let's say this is an A, this is an R, and this is a T. Okay. So these guys are exons. Okay, and these blue ones are introns. All right. And then, I don't know, let's make this an S. Okay. So what we can do, or what a cell can do, is, let's say this gene codes for A, this segment codes for R, this segment codes for T, and this segment codes for S. So just if we read this sort of like, in this direction, when we make this gene, what we can do is we splice out the introns. So we take the introns out, and what happens is we kind of curve it around. So you can kind of see that, where is it? You can kind of see it right here. So we have exon 1, exon 2. What happens is that we can, a spliceosome comes in, and it takes the intron and sort of bends it around and then cuts it and then connects the two exons together. Okay. So if we were to do that here, we could make a gene that would just be, that's the color I wanted. That would just be these segments that would spell A-R-T-S, spells arts. Okay. But we can also do it provides this we don't necessarily just splice out introns. We could splice out other things. We could move this thing around, so it allows us to maybe change the order of these genes. So what you might have is like a gene that has ARTS, 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 and then we can splice out different letters. So now we could change it around to where maybe we could spell rats. Okay, maybe we could spell we could go tarts or tars. Okay. So it provides flexibility we could cut out the introns. Okay. We could also cut out certain exons to get different gene combinations in a specific order. So what that allows is that even though we transcribed this one gene doesn't necessarily only give us one protein combination. We could have different protein combinations, which allows us to make different proteins from the same gene. That's why it's kind of hard for us to know what the entire human genome does, because we found that, it's not just as simple as one gene does one thing. One gene can do multiple different things, and it's hard to know what all those things are based on how the RNA is spliced. Okay, so this splicing is the RNA processing, and it has to go through this process where we splice the RNA, we put a cap and a tail on before it can leave the nucleus, and that's one way that we regulate what genes get activated and what genes don't get activated, or rather, what genes get expressed and what genes don't get expressed. Okay, so that's the first part. Is our splicing. That's the first way that we regulate genes. Okay, the other way that we regulate genes, which I think I've got in here we are. The other way that we regulate genes is with histones. Okay, now histones are these proteins that we basically are used to wrap DNA. So you can kind of see here that a histone are these little, light blue blobs, and we can basically wrap DNA around it twice. And what a histone allows us to do is it allows us to coil DNA and condense it into a much, much smaller space so that we can fit it all inside of a nucleus. But when you look at a histone, histones have these latches on them, right? These positively charged tails that latch the DNA on to keep the DNA together. Okay, and when the DNA needs to be read, these latches release, allowing the DNA to become loose so that they can be pulled apart, read, and then put back together. Okay, this is what is called acetylation. Okay, so acetylation is what we do is essentially we add these acetyl groups. Okay, and what that does is it takes our positively charged tails and it removes the positive charge, which means that these tails are no longer holding the DNA in place. Okay, so what acetylation does is it's modifying the histone to activate the DNA, to allow the DNA to be read so that we can go through transcription and translation. So acetylation is a way of turning genes on by allowing them to be read from the histone. Because if the histone is not on, if the histone is not activated, then there's no way to read this DNA because it's packed together. Okay, it's like if you're moving and you put all your stuff in a box, you can't do anything with it. You can't play with any of that stuff. You can't read anything in there. You can't do anything with it. You can't wear any of the clothes until you open the box. The box is closed right now, but if we open the box, now we're able to get and access all of that stuff that's in there. Okay. So that's the first way, is through acetylation. Okay. We also have what's called methylation. Okay. And methylation is where we take a methyl group, okay? Where's my picture of it? No. So what we do is we take a methyl group. Now a methyl group is CH3, okay? And what we're going to do with that is when we attach it onto, methyl groups want to attach onto the cysteine of our DNA, okay? And what that's going to do is it is going to deactivate certain genes, okay? Here we go. So we have our methyl group is CH3. What that's going to do is it's going to bind onto the cysteine of a DNA segment, okay? And it sort of acts as a roadblock because, remember, we have our RNA polymerase, and it's going to go through and it's going to read the DNA. But if we have a methyl group that's just chilling on the outside of our DNA, that sort of acts as a roadblock. The polymerase can't go forward because there's a methyl blocking its path. Okay, so what this does is it deactivates this gene because we can't transcribe it because there's something in our way. It's stopping us. Okay, so what these methyls do is they are a way to deactivate genes that would otherwise be activated. Okay, now what's interesting about these methyl groups is where do these things come from? Well, what we found is that the methyls are everywhere in your body, right? It's a pretty simple composition. It's just CH3. Okay. But what can cause this methylation is exterior sources, meaning your environment can actually cause your DNA to go through methylation and deactivate genes that would otherwise be activated. So what can cause methylation is an external stimulus, whether it be positive or negative, and that external stimulus can actually change your DNA. Okay. So this is a way that we can alter DNA by deactivating genes. So acetylation, we talked about activating genes. Methylation, we're deactivating genes. But what's interesting about methylation is that the thing that's causing the methylation is an external stimulus. So like the example we gave in class was these orange rats, right? Certain rats were given a different diet, which resulted in different methylation in the fat rat versus the non-fat rat. The other one was rats who were basically mothered well and were like groomed every day versus rats that were neglected, and the difference in their DNA because of the methylation. So we can see the difference in genes being activated and genes being not activated based on how their environment treats them. Okay, these rats were not groomed and resulted in the gene methylation and the gene not being active. These genes were groomed, genes active. These genes have low anxiety. These genes have high anxiety. Okay, so methylation is another way that we can deactivate certain genes based on the environment that they live in. Okay, the last way that we can talk about for eukaryotes is transcription factors. Okay, and a transcription factor is pretty simple. Okay, so we've got your promoter region right here, right? And I'm gonna slide this over a little bit. So what's interesting is that before your promoter region, you actually have another sequence called a regulatory process. So we could have a regulatory process like right here. That's really bad. Okay, so the regulatory process is, I mean, I drew it just to keep it on the screen, but this could be a large amount of space, right, in between the regulatory process and the actual promoter. So the regulatory process, what happens is it is the thing in eukaryotes that sort of activates the promoter and turns on the promoter, okay? But in order to sort of help with that, we have these things called transcription factors, and transcription factors are sort of like flags. So what they'll do is these transcription factors will bind to the regular. So we'll just say a transcript. So we've got these transcription factors, and what they're going to do is they're going to bind to the regulatory process and then bind to the promoter to sort of activate the promoter and get the promoter to bind here, right? And how it does that is pretty interesting, okay? Because it doesn't do it necessarily by activating the promoter. It does it by basically blocking all of the other genes, okay? So for example, right, imagine you're RNA polymerase and you're trying to find a gene to turn on, right? You're basically looking for any open gene, okay? So what these transcription factors are going to do is they're going to block every gene that is not the gene that we want on. So these transcription factors are a way to block specific genes and stop them from activating. Okay, so the way that's probably best to explain it is imagine if you're going in a parking lot, right? Like you're parking in the student parking lot. You have a specific space designed for you, and that space is designed by a number. Okay, you get to school late. You can only park in your space or in a specific space because every other space is filled, right? Or like, you know, if you ever go to like Six Flags or a football game or sporting event, something like that, right? They'll say, okay, you have a specific space that you can park in. You can park in space 275, and the way that you can't park in any other space because all those spaces are blocked by cones or whatever, right? These transcription factors are the equivalent of the cones in that parking. You can only park in your spot and in that one specific place because all the other places you could park are blocked by cones. These transcription factors are the equivalent to those cones, right? This RNA polymerase can only park and activate the one gene because all the other genes are blocked by these transcription factors, okay? So these transcription factors will bind to specific proteins that we're trying to keep off as a way of making sure that the RNA polymerase binds to the genes that we want on. So you can see here, these RNA polymerases are binding to genes A, B, and C because they're not being blocked by these different transcription factors, okay? So these are all the ways that we can modify eukaryotic genes. It's important to note, I don't know if I made this distinction, but So we've got two kinds. We've got prokaryotes and we've got eukaryotes. Okay, for prokaryotes, we talked about the tryptophan operon and the lac operon. Okay, those were the two ways that we can regulate prokaryotic cells, prokaryotic genes. For eukaryotes, we have acetylation, where we are modifying histones to activate certain genes. We have methylation, where we're modifying DNA to deactivate certain genes. And then we have transcription factors, where we are deactivating certain genes so that we are only activating the genes that we want active, okay? And the importance of both of these, of regulation, is that we only want certain genes on when we want them on, okay? We want and the reason why we want that is because every cell in your body has all of the DNA, right? If you take a skin cell, that skin cell has DNA for your heart and your lungs and your bones and your brain and all that other stuff, right? But we only want the cells that are responsible for your skin. And so we turn off all the other cells that have nothing to do with skin and leave on the cells that are skin genes. Conversely, in your brain cell, we turn off all the skin genes because we're trying to make brain cells, and we have the brain cell genes turned on. So we only want specific parts of the DNA on at specific times, and these regulatory ways are ways that we can do that. These are also ways that we can change DNA after the fact. Okay, so regulation of genes is important. And a lot of what we talked about in class was like what happens when these different regulation factors mess up, all the different diseases that can happen, the different disorders that can happen from regulating genes that should or shouldn't be regulated. Because there's a lot of diseases that result from misregulation of genes. And I'm not going to get into that now because you really don't need to know it for the AP exam. But you should know why we regulate those genes and the different methods for regulating those genes. Okay. That pretty much covers it in a nutshell, in a very, very quick nutshell. So yeah, we only have one more video to talk about, and that is Mendelian genetics. So having said that, I will see you guys next week.